Good afternoon, everybody. Welcome to the Coffee Science Seminar Series. And our speaker today will be Professor Mark Turner, and I'll introduce him fully in a few minutes. But if we go to the next slide, Mark. I'm Mike Gidley. I'm the Director of the Centre for Nutrition and Food Sciences. Mark is an affiliate professor in the centre, so a particular pleasure to host him this morning. The next slide, please. We acknowledge the traditional owners and their custodianship on the lands, the various lands on which we are meeting today. And we pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Okay. So today we have a 1 hour time slot. I'm hoping Mark will not take that whole 1 hour. And that there'll be plenty of time for questions and answers afterwards. So the important message today at the moment is that you can type your questions in. I will moderate the Q&A session. So you need to type your questions in the Q&A tab, not in the chat function. I won't be looking at the chat function. So please go into the Q&A tab if you want to ask Mark a question, and please feel free to do that during the seminar so that while the thought is with you, you put the question down. And then we will run through as many of those as we can at the end of the webinar. Okay, so let's move on to formally introduce Mark. So Mark is a professor in the School of Agriculture and Food Sciences and has got a specialty area in food microbiology, and that's what he's going to talk to us about today, particularly the work he's been doing on lactic acid bacteria. I think have you got is there a title slide next, Mark?
Yep.
Yeah, okay, so this is his title. So he's going to talk to us around lactic acid bacteria, the multiple roles that they can have for enhancing food safety, reducing spoilage, and to ferment plant-based dairy alternatives. Over to you, Mark.
Great, thank you, Mike. And I'll just minimize faces in the corner. Okay. Thanks, Mike. And I'd also like to acknowledge the traditional owners and their custodianship of the lands which we meet today, and also pay my respect to their ancestors and their descendants. So today I'm going to present some of my group's research in the food microbiology space. We're located in the Hartley-Tickle building in the beautiful in St. Lucia. And this is kind of research spanning over the last sort of 13 years. And been done in different areas and dribs and drabs over the years, and funding from different sources, including horticulture innovation and dairy innovation, ARC, and so on. But lactic acid bacteria really something I did my PhD on, and it's kind of a pet project that I've been able to luckily to maintain throughout this many years since that time. So just to take as an introduction, look at a little bit about food processing 101. For the non-food scientists out there. So a lot of the food processing industry do various food processing to make our food safe and also extend the shelf life and improve the quality of those foods. And these can involve a range of different things, such as physical processing methods. So we know about all these: heat, cooking, refrigeration. There's new high pressure foods we see in the market. There's radiation being used a lot for exported foods. We see chemical processing approaches, sanitizers like chlorine and peracetic acid being used on fresh produce to reduce microbial loads, and also preservatives being added to foods to stop or slow down microbial growth. So these are two well-known areas, but I guess the new kid on the block is a biological approach or a biocontrol approach, which I'll talk about a bit about today. And this is in the applications of food, is around using bacteria. Or bacteriophage, which are viruses that infect bacteria, using peptides from bacteria, which are antimicrobial, or simply just doing fermentation of foods to increase its preservative effect, improve the safety, and extend shelf life. So biological control is not a new approach to foods. It's been done for tens of thousands of years before people even knew what microbes were and could see them and understand what was happening. But we've got a huge array of fermented foods in the market, and about a third of all the foods that we eat actually undergo at least one fermentation step. So when we're looking at biological control, this is kind of the most basic approach, using fermentation for that. I'll talk a bit more specific uses of microbes as we go along, but applying this provides benefits. It's a natural process. It's got a perception from consumers of being safer. It can be a clean label when you're adding microbes to foods. Don't have chemicals and so on listed on the ingredients list. But it does have some challenges. So efficacy is a big one, where biological control doesn't have the efficacy or impact that physical or chemical methods will have. The impact on shelf life might be negative. There might be negative sensorial properties of the food. How do you apply the agent to the food? How do you apply the virus, the bacteriophage, or the microbe to the food? What's the cost involved with this? And lastly, what things about regulation and labeling? How do we approach that? And getting approval from FSANZ. Do we need approval? So this is a table kind of, I guess, showcasing the breadth of fermented foods out there. And I'm happy to report that in 2015, the periodic table of fermented foods was solved by Michael Gansler in Canada. It's kind of a tongue-in-cheek a slide or figure showing the different categories of fermented foods and grouping them together. Obviously, this is not all the fermented foods, there's thousands. This kind of gives a snapshot of the breadth. So we can kind of look at in the similar fermented foods in the vertical or the columns. So we've got wines over here, red and white wines. We've got ciders, beers, cereal-based fermentations, breads, soy-based fermentations, condiments, vegetable fermentation, sauerkraut, kimchi, and so on. Then we move over to the fermented dairy products here, yogurts, cheeses, and then the last column, I'm not quite sure you can see, is meats, salami, and so on. Over on the rows here, we've got spirits and vinegar shown here, grouped together, and tea, coffee, and chocolate, which are also undergo a fermentation step. There's a huge amount of information on here. It looks like someone vomited on the slide, but I guess the take-home point is, I guess we'll just get you to focus on the colors. And you see there's a color coding all throughout this. Every box has a color in there, or most do. You see that the green color is the most common, followed by the red, followed by the blue, followed by the yellow. So green indicates that lactic acid bacteria are involved. And that's why I'm giving this talk, because we work on that area. So LABs, or lactic acid bacteria, are really the most commonly used organisms in fermented foods, followed by yeasts. Obviously with wine and beer and bread and so on, yeasts are very important. Next, followed by molds, we see in blue, particularly sort of the mold ripened cheeses and chocolate and so on down here. And then lastly, the acetic acid bacteria. So obviously in vinegar, but also in chocolate and a few others. The lactic acid bacteria, this is some examples shown at the bottom. I might just move to some new technology, show you there's some examples of some of the genera of lactic acid bacteria. So lactobacillus, lactococcus, and so on. So let's look a little bit about LABs, or lactic acid bacteria. So they're obviously in a huge range of fermented foods. So they've got to generally recognize the safe status. A lot of them, certain species or certain strains are actually listed as probiotics as well, but there's a difference between fermented foods and probiotics. A lot of them produce antimicrobial compounds, and that's what gives those fermented foods the extension and shelf life. So organic acids, lactic acid, as the name of these organisms suggests, but other ones produce antimicrobial peptides, hydrogen peroxide, and a range of other small chemicals. They produce flavor compounds and affect the texture of foods, which is important in making those fermented foods taste better. And they're naturally present on fresh foods as well. So if we look at any sort of fruits, vegetables, meats, and we take a sample of the surface of that, we'll find lactic acid bacteria in most cases. This can be 1,000 to 10,000 per serving of these fresh foods. So this really provides a rich source of new lactic acid bacteria that we can obtain and screen for applications. And that's what we've done in our research group. So we're looking at these plant-derived lactic acid bacteria. And this was based on a project I had back in 2009 from Hort Innovation, where we proposed that we'd isolate good lactic acid bacteria from fruits and vegetables, with the idea that we could then reapply those back onto these fresh produce to enhance the safety of that. This was on the back of some outbreaks with salmonella on fresh produce, and the vegetable industry were very interested in that, and that's what kickstarted our research in this space. So we're interested in isolating lactic acid bacteria from a wide range of fruits, vegetables, and make a collection that we could then use to test for applications. So our applications, we're looking at controlling Listeria monocytogenes on ready-to-eat fresh produce. Listeria monocytogenes is a foodborne pathogen. It caused probably the last big outbreak in Australia was 2018 with a rock melon outbreak. About 21 people were sick and 7 deaths with Listeria contaminated rock melons. It's an issue for obviously any food that is comes from the farm can have Listeria on it. It's an environmental sourced organism that can get into our food supply. We're also interested in ready-to-eat sliced ham because Listeria can be a problem in deli meats as well and has caused outbreaks in the past. So this is a safety application using these LABs to make these foods safer against Listeria. Then we were also interested in, and this is from Nidhi Bansal, an idea that she proposed once we started doing this work, was could we then use these bacteria to screen for inhibition of mold on cheese. So this is a quality application to inhibit the growth of fungi on cheese and extend the shelf life. And then more recently, we've looked at these bacteria to carry out fermentations of plant-based dairy alternatives. So I'll cover each of these sections as we go through. The biocontrol areas are around the anti-Listeria and anti-mold activity, and we basically have a collection now of about 900 isolates. This is from 10 years ago, from fruits and vegetables, from all these different areas. And these were great student projects where students would go to the supermarket or farms or markets and so on and bring in these fruits and veggies, and we'd isolate LABs. And then we'd screen those against Listeria and Penicillium using a simple spot assay. We'd look for inhibition zones around the LAB colony, indicating that it's inhibiting that target organism. And then we'd identify what those were and further characterize them, and then do some challenge trials on the food. So this is a bioprospecting approach. And so we got those and we started the screening. So this is an example of a plate where we had Listeria monocytogenes in the agar. So that's the cloudy appearance in the background. And then we spot on our lactic acid bacteria isolates here from the fruits and vegetables. And what we see here is you see some of them don't have any activity around the colony. Some have very small activity. There's a small clearing zone indicating the bacteria is producing something. And then other ones have quite large inhibitory activity, where this bacteria is producing something that's either inhibiting the growth or killing the Listeria in the agar throughout the agar there. When we did the screen of 900 isolates, we found the best ones against Listeria were Lactococcus lactis, and we'll further characterize them in a minute. When we screened against the fungus, so this is the mold growing across the plate. This is a Penicillium communae, which is a common cheese mold, spoilage mold. We have 3 spots of LABs here. These 2 are not very good, whereas this one here is quite good. It's able to inhibit the growth of the mold around the colony. And what we found was all of these were Lactobacillus plantarum that had the activity out of 900 isolates. We got about 12. So that was good. We've got some new potentially promising strains that could be used in food safety and quality. So we looked at the Lactococcus first, looking at the anti-Listeria activity and whether this was due to nisin, which is an approved food preservative it's used as an additive in the food industry for a few decades now from Lactococcus. So these strains naturally produce this in the environment when they grow on the plants and fruits and vegetables as a sort of a competitive, it gives it a competitive advantage. So we're interested are these strains nisin producers, and from PCR we could show that they contain the nisin gene. This was a negative strain. It's a negative control. Nisin's an antimicrobial peptide, 28 amino acids long, it targets other bacteria like Listeria monocytogenes. However, when we did some genome sequencing, we identified another peptide bacteriocin gene cluster as well, Lactococcus 972. So there was a question whether which is actually causing the antimicrobial activity. So we're interested in mechanisms of action, understanding this well. So what we did here was to make a gene mutant library. So we introduced a plasmid into one of the strains that produced the antimicrobial activity and made a random transposon mutant library. And then again, went back and screened 5,000 colonies from that mutant library from the one strain. And you can see an example of the plate here. This is the spotted mutants. And you can see the clearing zone around the colony, indicating this is the target organism in the background. And you see they're all producing large zones, except for this one here, it's producing no zone. So it's lost the ability to kill that Listeria. We screened 5,000, we got 8 mutants, and all 8 mutants contain a transposon insertion in the nisin gene operon, which is a very large operon consisting of several genes, which strongly suggests that nisin is the key antimicrobial here. And when we put those mutants on the plate, we completely lost activity as well. So this seems to be the main one on agar plates. But then we wanted to test that in food trials as well. Is nisin the only thing that's actually going to inhibit that Listeria on foods when we run trials? So we put some cells on cut iceberg lettuce along with Listeria cells, and this is the number of Listeria cells here. And what we see, this is an abuse temperature here when we were initially playing with this method. So over 7 days, the Listeria is able to increase in numbers without any lactococcus there. When we add lactococcus, we get a slight reduction here and some inhibition of growth at 3 days compared to the control, which doesn't have lactococcus. But by 7 days, the Listeria had grown up to pretty similar levels as well. So we don't really get killing effect here. So what we thought we'd do was to use cells plus supernatant, which is what we call a fermentate. So get the bacteria to produce whatever the antimicrobial is, and then apply that as a mixture. So cells plus whatever the antimicrobial is, and then put that onto the lettuce and run the trials there. So we ended up reducing the temperature down to more realistic temperatures here. And the Listeria actually numbers in the black bars here reduce over time about 1 log, which is good in cut iceberg lettuce without anything in there. And what we found is when we put the fermentate from the wild type strain, we get quite a large killing effect and undetectable levels of Listeria on day 0 immediately after the application. And we never saw any Listeria again over the 10 days trial. And we tested our mutant as well. So the mutant that's basically mutated in one gene that's lost the ability to produce nisin, that's the only difference. What we found, it didn't have any killing effect. It did have a slight inhibition or greater reduction in numbers of Listeria over time. But what we can say from this is clearly that nisin is by far the major antimicrobial in this assay against Listeria. We also tested on ready-to-eat sliced ham. So we ran the trials again with our wild type and mutant, and no lactococcus in there as well. And these all have Listeria. So in ham at 4 degrees Celsius, the Listeria in black bars here is able to increase in numbers over 10 days. When we added the fermentate, we're able to get reduction then to undetectable levels. And that remained over the 10-day period. This is with the nisin producing lactococcus. In the nisin mutant, we didn't get that reduction here, but we did get growth inhibition. So it didn't increase like it did without the lactococcus. So it suggests here that nisin again is the major killing factor here, but there is other things in the fermentate, probably organic acid, that is able to inhibit the growth of that Listeria over the shelf life. So that's the work on Listeria. The fungus work here is shown. So I had an honours student who did the screen against the penicillium mold, and then we wanted to do a challenge trial on some cheese, and obviously we weren't going to make cheese and ripen cheese. It takes a lot of effort. I'll present some work that we did later on after that. But we used cottage cheese as a model system where we inoculated in the lactic acid bacteria, the Lactobacillus plantarum that we got from the screen with the penicillium communae, and then stored that cottage cheese over time. And we put that in just little blobs of cottage cheese on Petri plates. And you can see without the lactic acid bacteria, just with the mold inoculated, after 4 days, you see there's a lot of furriness on that cottage cheese, and looks pretty funky by day 12. Some of the LABs that didn't have activity on the agar plates, we put them in sort of as negative controls. This is a Weissella, Lactococcus, and a Leuconostoc. And you can see there was a little bit of inhibition of mold for some of them, but you could still see there was mold spots all over it. And by day 16, it looked pretty bad. The Lactobacillus plantarum isolates that we got, when we put those in there with the mold, even by day 20, we still didn't see any mold spots coming up there. So it's quite a large improvement in the mold inhibition activity there. So this was really positive, and from those screens that we did, the Lactococcus has anti-listeria activity here, but obviously the activity that these organisms have is very specific towards the target pathogen. They don't have this broad spectrum. So whatever we screen the LABs against, we get different organisms coming out most of the time. So that's with cottage cheese, which is not, which mold spoilage is an issue, but it's dealt with pretty well. So we wanted to then move on to cheddar cheese and actually use the Lactobacillus plantarum as an adjunct culture and actually make cheddar cheese. So adding it in the milk at the first step, and Nidhi Bansal and Elizabeth did most of this work. So we made some cheddar cheese, ripened it for 24 weeks. We shredded that then, and then added in some Penicillium communae mold and looked at the cheeses on day 4 and 7. And these are day 4 results. They all look pretty good. This is the control without Lactobacillus plantarum. These 4 here contain 4 different strains of plantarum in there. By day 7, you can see the control cheese is looking pretty furry. The mold is growing, and also on this one here with this plantarum strain. But these three cheeses here look pretty good. There's a little mold spot there and there, but largely it was improved compared to the control. This plantarum strain didn't grow very well in the initial fermentation phase. The numbers are about 1 log less than these ones, so that's probably why it didn't work as well. So that's with when we add in mold, we added pretty high levels, which is probably a little bit unrealistic, but just to speed the assay up. When we just shredded the cheese and then exposed it to aerial contamination in the lab, and there's molds in the air that fly in and land on in our experiments, we then held those cheeses for different time points. So day 6, they all look pretty good. But then by day 35, and so it took longer for the mold to grow because the inoculation was less. The inoculation level was lower. The control was pretty furry, so was 276 plantarum. But these 3 other strains of plantarum, when added in the cheese, were able to still resist the growth of that natural mold contamination. This 299V strain here we actually got this from a probiotic product. You can buy this in the supermarkets. So not only have we got a anti-fungal cheese here, we've got a probiotic cheese that has high numbers of probiotics. So we've got a win-win situation. We've just recently submitted this work for publication. So that's kind of some biocontrol research that we've done. I'll move on to more recent work where we've looked at using some of these plant LABs and doing screens to see whether we can find strains that might be useful in plant-based dairy alternatives. So this is a project that's sort of a 2 PhD project that was initiated between UQ and DTU, Denmark Technical University. And this came about a couple of years ago, where UQ and DTU had an agreement where they were going to try to collaborate more in research and in the research training space. And they put up scholarships where if you have a project that's a collaborative project, UQ will put a scholarship and DTU will put a scholarship, and you have 2 PhD students co-supervised as a team, and they'll be able to cross over and visit the other institution. So in DTU, Klaus is the supervisor, Anders is the PhD student. At UQ, we've got Wang Kang, who's been going for nearly a year now, who's our PhD student, and Sangeeta and I'm the primary supervisors of Wang Kang. So let's just look at plant-based foods. It's all the rage now, and why not join? So obviously grown in popularity in recent years, greater health awareness, more veganism. There's concerns about animal welfare, environmental impacts, and also in dairy foods with allergies, lactose intolerant, and so on. So there's a huge, wide variety of plant-based foods coming out on the market now. It's really exploding. The plant-based milk alternative area is also exploding. So the market is around $27 billion internationally, and it makes up about 15% of the cow milk when you compare it to the cow milk market. So dairy-free milks, and I'm not going to get into whether it should be called the milk or something else. That's for another day. But there's a huge array of these products out there, and some examples are shown in this picture. We were interested in starting this project. We wanted to look at plant-based fermentations and develop. We were interested in, we needed to find a model system, and we ended up choosing Almond to start the project with. And because almonds are a big deal in Australia, so the market share for milks in Australia between 2015 and 2020, soy milk has reduced its market share from 69 to 48, whereas almond milk has increased from 18% to 44% of these plant-based milks. And now it's on par or even higher than soy milk now. This data is a little bit old. Almonds are a large industry in Australia, we're the second largest producer in the world after the US, but we only produce 8% of what they do. Most of theirs is in California. So they're really the main one by a long way. But even though we're only 8%, this value is about $1.6 billion, and we export about $550 million worth of almonds. And it's Australia's most valuable horticultural export. So we were interested in looking at fermentation of almond milks to potentially enhance the flavour and texture of almond plant-based milk alternatives. And there are some, almond yogurts and so on, but we were interested in possibly making cheeses as well, and just generating new products through fermentation. The issue with fermentation is that dairy fermentations are well characterized, and there's lots of strains that are being used by the dairy industry. But however, typical cheese-making Lactococcus strains that are used in dairy industry are unable to acidify pure almond milk. So when we got one of our dairy strains, because we've done a lot of work on starter cultures in the dairy industry, we put some of them into almond milk, and we monitored the pH over time. And this is one of the strains. When we put it into cow milk, the pH falls quite well. They're able to acidify that milk, produce lactic acid. But when we put that strain into almond milk, the blue line here shows that it doesn't produce any acid in that product. It's unable to grow. So we can't just use the typical dairy starter cultures for these plant-based milks. The yogurt starter cultures are used, and they're partially okay, but they're still not very efficient acidifiers of almond milk. They're not Lactococcus, they're other bacteria. So we were interested in trying to find some of our plant-based lactic acid bacteria that could be better at fermenting some of these plant-based substrates. So a lot of these culture suppliers are interested in fermentation of plant-based, but what they'll do is suggest adding glucose into that dairy alternative product so that the microbe can utilize the glucose and produce the acid. So instead of that, we wanted the organism to actually use the substrate that was naturally present there rather than adding sugar on top of that, because then there'll be residual sugar there that's not touched. So we went back to our collection 10 years after we made it, and wanted to ask the question, are plant-based lactic acid bacteria better at acidifying plant-based dairy alternatives? So again, our collection, and we did acidification screening in almond milk that we bought. And 10 years later, a few of those strains had died in the freezer or appeared contaminated, so we didn't really worry about them. But we managed to get 600 out of that collection from about 900. And we inoculated those individually into unsweetened almond milk that doesn't have any additional sugar. And we added a pH color indicator in there, bromocrystal purple, which changes color. So this is the normal almond milk. It's kind of gives a blue purple color. And when the pH reduces, you get a yellow color here indicating acid production. So it's a very visual screen. It works quite well. And we also measured pH, and just to verify this was correct, and also measured final pH after 24 hours of a number of strains, around 200 here. And you can see these are different strains put in almond milk, and you see this spectrum of activity. Some are completely unable to produce acid, and some are able to produce moderate amounts of acid, and then other ones are able to produce high amounts of acid down here. If we look at their acidification rates, this is sort of endpoint here, but if we monitor the pH over time, we see this strain here is very fast at producing acid in almond milk. This strain is intermediate activity, and this strain up here is poor, and this is just the uninoculated one here. So we were quite interested in knowing, okay, from our library here, we don't really know what, we have some idea of the organisms there. We don't know all of them. We haven't identified all of them. This is sort of a blind screen. So we wanted to know what's the difference between these guys down here and these ones up here, for example, or these ones up here. So we obviously thought that, the sugar that's present is probably a key factor, as it is a substrate for the lactic acid that's produced. When we look at almond milk sugar composition, it's 88% sucrose, 6% glucose, 6% fructose. Very different to milk, which is all lactose. In sucrose, a disaccharide, glucose and fructose. In lactic acid bacteria, so it's a well-known pathway to produce lactic acid. Transports the sucrose, cleaves it into the monomers, and then these enter glycolysis here. We did whole genome sequencing of our strains. We got around 12 strains that were really strong acidifying. And the sucrose genes are clustered together in the genome, where you've got the transporter, the hydrolase enzyme, and the phosphorylation enzyme here, along with the gene regulator here. So all our strains had this gene cluster that could acidify almond milk. But we wanted to know, I guess, so we can infer that that's important, but we wanted to know really in detail if we lost this sucrose metabolism in this strain, are these strains, is a mutant then still able to acidify that almond milk, to really confirm that, yes, sucrose metabolism is the key property that allows almond milk acidification. So again, we love screens in my lab. We've got lots of students floating around that love monotonous, boring experiments sometimes, but this was done by actually a PhD student, Wen Kang, and he was very persistent. He screened 50,000 colonies on a sucrose agar with a pH indicator. So the same pH indicator we use for the almond milk screen, but you see this purple background here. And when you get acid production, the colonies appear yellow, indicating that they can use sucrose. So Wen Kang would have done about 200 plates of 250 colonies per plate. This is the wild type strain, so it's definitely fermenting sucrose. And out of those 50,000 colonies that he screened, he got to about 30,000 and still hadn't found one that had lost this ability. So we're looking for ones that now are no longer yellow; they've turned purple, indicating they've lost the ability to ferment sucrose. He got to 30,000 without success, but then he managed to pick up about 6 in the last 20,000. And one of them shown here. So if you streak out this negative strain here, you can see that it's not yellow. And the colonies are a little bit smaller as well because they're not fermenting the sucrose. There is some residual glucose in this media, so the bacteria can still grow. Otherwise, it wouldn't be able to grow at all. So we did genome sequencing of those 6 negative strains. So this is kind of a spontaneous mutation approach. We actually try to knock out the gene for 3 months, deliberately using a GM technology. With no success, so we thought we'd go old school and actually just plate colonies on plates and do a visual screen. It ended up working though, so we did whole genome sequencing on these 6 isolates, and for each 6 we only saw 1 mutation for each, and we saw the only mutation we saw was in the sucrose operon, the sucrose gene cluster here. So we saw frame shift mutations in 3 different spots, and 3 of the mutants were in the same spot as well. So 1 base pair deletion, 14 base pair deletion, 1 base pair deletion. So these would actually disrupt the activity of these enzymes, or the transporter here, and also the hydrolase, which means that I'm unable to transport in the sucrose or cleave that sucrose, which agrees with why it's negative on these plates. And we've shown that with growth curves and so on. So we've got our sucrose negative mutant. Now the question is, if you put it back into almond milk, can it still ferment that almond milk, or has it lost that ability? So we're testing a few different plant-based milks. So we wanted to pick a few that were rich in sucrose, so almond, cashew, coconut, and also we've done macadamia as well. These are majority or all sucrose. We also did a few other ones, rice milk, oat milk, cow milk, which are 0% sucrose, to see whether we would expect it wouldn't have any effect. So the results are shown down here. This is the endpoint pH level of the different milks. So these are the sucrose-rich milks here. This is the wild type strain, which acidifies very well. This is the starting level, the black bars here, and this is our mutant here in the triangle. So the sucrose transporter mutant basically is unable to acidify almond milk or cashew milk. Coconut milk, it reduced it a little bit, but we think that coconut milk had some other sugar in there as a commercial source, but we need to double check that. But you can see where the sucrose is present in that media. Obviously, losing that sucrose metabolism is critical, and basically halts fermentation. In the other plant-based milks, rice, oat, cow, we don't see any difference between the wild type and the mutant for each of these. So sucrose metabolism doesn't play any role as expected. For the cow milk, we actually find the plant lactic acid bacteria are really bad fermenters of that. So the pH only dropped to, high 5s at that point, and largely because they don't contain the lactose metabolism genes. And we've confirmed that through whole genome sequencing. So putting your dairy strains into almond milk doesn't work very well because it's mainly sucrose over here, but also putting your plant strains into cow milk doesn't work very well as well. Okay, that pretty much wraps up our data. I'm just going to present a little bit on other milk alternatives and something we might have heard about, a little bit detached from what I've been talking about, but this is around precision fermentation. So if we wanted to produce milk, we can do it, go back to square one and produce the actual milk itself. There's a lot of interest in this at the moment with producing the proteins in milk, casein and whey proteins by microbes, genetically modified microbes. So you can actually produce something that's like milk to then make cheese and so on. There's a huge interest in this area. The US Perfect Day has been around for 10 years and had hundreds of millions of dollars thrown at it recently to further develop this. Eden Brewers in Australia, and they're I think part of the Trailblazer that just recently got announced, and also precision fermentation is a large part of that Trailblazer project that's just been announced as well. So this is an area that you'll hear a lot more about in the next few years with UQ being involved. So this is a nice little amusing picture where in the old days you would feed a cow some grass and then milk it and get your milk and then make it into cheese. But now we're using plants to put into a bioreactor or a stainless steel cow to feed those genetically modified microbes to produce casein, and then we can make our cheese with that microbe-derived casein protein. So there's a range of advantages with this, with water use, land, energy, and feedstock. And Perfect Day are probably the most advanced with they're producing powders, protein powders, and so on, and putting their products into alternative foods like this ice cream that they're marketing at the moment. So just in conclusion, so from the work we've done, so lactic acid bacteria fermentate, have potential as biocontrol agents to control listeria on lettuce and ham. So you could apply this post-wash steps just before you package up this food, provide an extra safety hurdle in the event of a rare contamination event. So as shown here, you could apply this to a processing line where you spray this mixture onto the product at the end. And this would ultimately improve food safety and also reduce the chance of food recalls and food waste and so on. LABs have the potential as anti-fungal applications in cheese to reduce spoilage. So we've got some really nice results there showing we're able to inhibit molds, naturally contaminating molds in cheeses. And then lastly, plant-derived LABs provide new opportunities as starter cultures to ferment dairy alternative plant-based foods. So there's a lot of people to acknowledge, a lot of students and staff that have been along for the ride with these projects over the years. And I'll also acknowledge Hoyt Innovation for funding several projects, and also the ARC Dairy Innovation Hub and ARC for funding that, and also our collaborators at DTU. So thanks for your attention, everyone. I'm happy to answer any questions, and I'll hand back to Mike.
Okay, thanks very much, Mark. Fascinating story. It's nice to see it all connected up like that. That's tremendous. So we have a few questions in the Q&A. I think I'll start off with quite a general one. So early on in the talk, you mentioned that the LAB are generally recognized as safe, GRAS. So there's a question do you know of the 900 LAB you've screened, whether those are all GRAS, or how would you know what is GRAS and what is not GRAS?
Yeah, good question, Mike. So we've probably identified probably 150 of those, and I would say most of them are, probably 85% genus that we recognize and you know, are already used in food fermentations or various applications in foods. There are some of those that we do see that we're, you know, things like Enterococcus we do get coming off, and they are also used in food fermentations, you know, meat fermentations and so on, but they're kind of a little bit on the edge of, you know, the safety and so on. Sometimes they carry antibiotic resistance genes and so on. And we do also see a few other bacillus and a few other random ones popping up in that collection. So when we do the screen, the first thing we do is then go and identify the ones we get out of that. And then what we'll do is typically, you know, avoid those ones which are probably going to be harder to push further into applications. Yeah.
Maybe if you could explain just a little bit about what it is that determines GRAS status, what are the criteria so that people understand that?
Yeah, good question. I don't know the perfect answer. I think they sort of have a long history of safe consumption, but how do you get to that long history of safe consumption? I'm not sure. The European Union have a list of sort of pre-approved organisms that are used in, you know, have been used in fermentations and so on. They have this QPS status, and the USA have the GRAS status as well. But largely it's based on sort of, you know, there hasn't been, you know, hasn't killed people. You know, it's been used in fermentations or whatever else. Yeah, a history of safe consumption. I mean, with immunocompromised people, these organisms can get in and actually can cause issues. So they're not safe for everyone, I guess. But it's very, very, very rare that it will actually cause, you know, endocarditis in someone that's, you know, they've generally got, you know, very low immune system, and something else happened. There's, you know.
Next question is from Xiao Yang, and it's about the use of LABs in the wine area and whether that could be extrapolated to other beverages as well. So he notes that some strains produce acetaldehyde and with anthocyanin can enhance and stabilize color after malolactic fermentation in wine. Do you know, is that concept used in non-alcoholic beverages as far as you know?
I'm not sure. So yeah, there is. Yeah, I know that we've done some work before on flavor formation with lactic acid bacteria, and LABs are pretty important for that secondary fermentation in wine production as well. Yeah, I'm not sure. I mean, wine, we haven't done work on wine. We've done some work on flavor, and there's other areas as well, like texture and so on, that they can help. Improve the, you know, the quality of some foods. But I think, yeah, the secondary fermentation in wine is Onococcus is the one that's involved there. That's kind of commonly used actually, but in other foods, it's not so much.
Yeah, Xiaoyang notes that Lactobacillus plantarum is also becoming used in the malolactic fermentation. It sounds like an interesting discussion you might have with him.
Yeah, I haven't heard of that. I know about Onococcus. That's kind of the wine LAB that everyone talks about, but not Lactobacillus. I think Lactobacillus plantarum is one of those broad sort of, you'll find it almost everywhere when you look in a lot of different areas. So it's kind of quite versatile. So it probably doesn't surprise me too much.
Okay. Another question from the more like, I guess, the challenge in food safety around listeria. The question from John Dixon around, does the frequency, how does that reflect the type of farming system, i.e. sort of the proximity of livestock, irrigation, humidity, dry land, and so on? Is that well understood?
To a degree it is, yeah. So, well, that the rock melon outbreak that happened years ago was probably a combination of weather events where there was a lot of wind and a lot of dust, and it probably picked up, the listeria is commonly found in soil. So it picked up the listeria from the soil and blew it onto the rock melons. And then rock melon has a very netted skin, so it's very hard to remove organisms from that. And so that was probably an environmental, you know, I guess a extreme environmental situation that resulted in contamination of that. They have found that things like silage can have listeria present in there. Animals can carry and shed listeria in their feces as well. So anywhere you look, listeria is pretty much there. So it's one of those organisms that it can be a massive issue in trying to eradicate from foods.
Sure, and will continue to be so, I think. So on a sort of similar vein from Pat Blackall, he notes that food production systems are very cost aware. And so what can you say about the cost of using lactobacilli as an additive for safety? Are they competitive with chemical treatment alternatives?
Yeah, they won't be, but we've done some work in some projects where we costed this, and we estimated sort of a bag of salad, if we applied these Biocontrol to control Listeria, then it would be around 10 cents per bag, or 200-gram bag of salad mix. So whether that's prohibitive, it's probably 10 times more expensive than, say, a chemical preservative, peracetic acid or chlorine or something. But it has that advantage of sort of a clean green, no chemical added, and the veggie industry were really interested in this from that perspective, trying to remove the chemicals out of their process. Yep.
Okay, I think moving on to the final section, you talked around precision fermentation. So first question from Shinla is around, do we have suitable reactors in UQ for that growing precision fermentation need?
You'll probably answer this better than I do, Mike. I'm pretty sure it's no. I suspect that the Trailblazer will massively build that capability, because it obviously needs to be in large scale to make that feasible. You can't just run these in 20-litre bioreactors and so on. I think they're planning on building a pilot plant. Is that correct, Mike?
Yeah, so there will be sort of laboratory scale, sufficient to do the screening and sufficient to generate enough samples to do taste tests and things like that. To understand functionality, and there will be a larger scale facility to be built in Mackay with our partners in QUT, which is expanding on a current capability there, which is basically making use of the proximity of the sugar industry there and using sugar as the primary the carbon source for the growth of these microbes. So that will be at full production scale, so that people can really test this out at the scale that they would need to. So yeah, as Mark says, this will be an important part of the Trailblazer process over the next 4 to 5 years. And then relating to that from Barbara Williams, for vegan cheeses, the protein may come from fermentation. What about the fat? Because clearly cheese needs fat as well to have its properties. Where would that come from?
Come from a plant-based source, I presume. Yeah, that's one of the questions. I mean, milk is not as simple as just producing casein or whey. There's different forms, and they're in micelles and different structures and all this sort of stuff. So I guess that's one of the massive challenges that faces, is trying to recreate the complexity of milk. It's not as simple as just producing. And then you've got these other things, do you add lactose, and do you add a different sugar, and how does that impact the final product and all this sort of stuff? So it's early days still, Barbara, unfortunately, but we'll get there.
I guess I'll note also that's a project area in Trailblazer as well. One of our partner companies is exactly addressing that opportunity of using tailored microorganisms to precision fermentation to produce fats that could then be combined with fermented products for proteins to create protein plus fat combinations that address cheese, meat, other areas where both protein and fat are needed for the sensorial response. There's no other questions in the Q&A, but I've got one myself, Mark. You mentioned that LABs are quite often also associated with probiotic activities for humans. I guess my question is, how connected is the antimicrobial usage to their potential for benefit in the human microbiome? In other words, is any of the antibacterial potential of LABs, how important is that in tailoring our own microbiome?
Yeah, good question. I mean, there has been some studies looking at antimicrobial, specifically for the Listeria activity, and people I've seen studies published where they've used strains that produce bacteriocins, and they can, in animal models, they'll actually prevent Listeria infections and so on. In humans, though, there hasn't been a huge area of, that's kind of one of the probiotic benefits, that it has antimicrobial activity. But really whether that has an impact in a human. You know, it's hard to run those experiments where you give people probiotics and then try to infect them. But in animal models, they have shown some benefit with this against Listeria in animal systems using LABs that produce antibacterial compounds. A lot of the probiotic stuff is around more so, I guess, stimulating the immune system and inflammation, all these other areas. And then the real push at the moment with probiotics is using these microbiome-derived probiotics. So, you know, isolating them from microbiome studies, and you know, the fecal transplant is obviously the crude way to do it, but actually picking out individual species and restoring them through the inherent organisms there. These LABs are present in the small intestine, but they're not really dominating in the large, so they're kind of yeah.
I guess the more we learn about the molecular mechanisms involved, the active components and so on, then the easier that sort of screening will get. So that, as you say, we're not going to infect people deliberately to do a challenge test like that, but we could screen for products that are known to be antimicrobial. I guess at least that's a less ethically challenged area.
Yeah.
Okay, I think that we come to the end of the Q&A. So thank you all for the questions, a really interesting and diverse set of questions, which goes with an interesting diverse talk. So I think that matched very well. So just two things to do first. First of all, to thank Mark again for what was a wonderful tour through his research in the area of lactic acid bacteria and their diverse applications in foods. And secondly, to say, come back in a week's time, please, and hear from Dongzhe Zhao, who's going to talk to us around how to use 3D characterization of crop water use to understand routing systems in the field. So a very different topic, but also incredibly important in these days of uncertain amounts of water in the soil, from at the moment too much to other times too little. So please tune in again this time next week, and you'll hear about that as well. So finally, thanks very much, Mark, and thanks to everyone, and have a good day.
Thanks, Mark. Thanks, Mark.
Thank you.